Now, I'm excited to introduce Dan Bradford. Dan is trained as a molecular biologist, started out developing genotyping assays for paddlefish and validating microarray data. He has spent the last 13 years working with laboratory automation systems to develop high-throughput genetic screens. Take it away, Dan.
Okay. Hi, everyone. I'm Dan. I'd like to thank Formulatrix for giving me an opportunity to present our work here. I'm going to talk about a high-throughput screen we've developed with automated liquid handlers to develop the knockdown of genes in the process of tissue. Getting a little bit of slides. There we go. I manage an at the Stowers Institute for Medical Research in Missouri. Our institute was founded in 1994 and began research in the year 2000. Now, Stowers is a research institute. Our founder, Jim, also founded American and left a significant part of his fortune as our endowment. We have about 500 institute members in 21 research labs spread across core labs and administrative positions. The focus and strength of our institute is the collaborations between core facilities and research labs. When you come here and do your work, you don't have to be an expert in every single discipline. As a result of this, papers published here often have long authorships. So within the molecular biology core facility here, we have 3 teams. Each has a different focus. The next-gen and Sanger sequencing team, the genome editing team, and the automation and PCR technology team, which I manage. Our mission is to support the 21 research labs here through provided services, collaborative projects, as well as independent research and development of emerging technologies. I've been working in automation here since 2006 and took over management of this group in 2012. We collaborate with research labs here to develop and lead high-throughput automated projects with a variety of lab robots. We have 3 TCANs, 1 biomech, a QPix colony picker, a yeast grading robot, and a variety of high-throughput analysis instruments that enable us to successfully complete these projects and screens. Over the years, our group has contributed to 15 publications, of which the most prominent are these 2 papers in Nature and a third in Cell. We also support qPCR, which utilizes an additional 2 liquid handling robots, as well as droplet digital PCR, which incorporates liquid handling, microfluidic droplet generation, a thermal cycler, and a flow cytometer that reads the droplets. To help describe how the RNAi pathway works, I'm going to do a very brief review of molecular biology's central dogma, in which DNA is transcribed into RNA and subsequently translated into protein. That's it. End of review. At Stowers, we perform basic research, which is to say that our focus is to determine what genes and their protein products do. One way to do that is to knock out or knock down gene function and observe the effects on phenotype. RNA interference uses an already existing cellular pathway to knock down expression of specific genes. When double-stranded RNA enters a cell, a surveillance protein complex called dicer, which isn't shown here, detects the presence of double-stranded RNA and cleaves it into small molecules. At that point, the RISC protein complex binds the small single-stranded RNA molecules and presents them in a way that, due to the complementary base pairing properties of nucleic acids, can only bind to specific RNAs with a complementary sequence and targets them for destruction. As a result, the messenger RNAs are destroyed, which inhibits the translation of the protein product. And the model organism that we are using to investigate RNA interference is the Planaria Schmidtia mediterranea. Our particular colony came from a water fountain in Spain. It was actually lost once in the past prior to our collaborators' move to Stowers due to filtration issues, and the lab had to return to the source to reestablish it. However, this fountain has since been removed, so ours may be the last living colony from there. Planaria are an ideal model organism to study tissue regeneration due to the abundant pluripotent stem cells called neoblasts present within them. As a result, these organisms can be dissected in a variety of ways, and they can regenerate back into a complete organism within a matter of days. If we can fully understand and harness this regenerative process, this could have very profound implications on regenerative medicine. So I'd like to give a little background here so you can appreciate the evolution of this type of screen. Early screens simply injected double-stranded RNA directly into the tissue. This produced a localized and a temporal effect. Our collaborators then moved on to plasmid expression vectors, but had to abandon gateway technology simply due to its high cost. They then developed ligation-independent cloning, which entailed doing PCR on cDNA, where the 5' regions would have restriction enzyme sites. This required cleaning up the PCR reactions to remove the primer dimers and unincorporated DNTPs, a restriction digest, a T4 polymerase reaction to add sticky ends, and then these inserts were added along with the vectors into the transformation reaction, and you would hope that once they got inside the E. coli that its own repair mechanisms could produce a fully intact RNAi expression vector. Modern screens now use the soft serve technique in which these transformed E. coli are mixed with beef liver and then fed to the planaria. The worms are almost all gastrointestinal cavity, so the RNAi vector is delivered very uniformly throughout the organism, which is shown here by the worms having ingested beef liver along with blue and green food coloring respectively. Now, the drawbacks to this method are it's really labor intensive, and something that's labor intensive isn't necessarily easy to automate with robots. It's also got a high reagent cost due to the cost of restriction enzymes and T4 polymerase, and our efficiency of transformation just was never very high. So that meant lots of picking and screening. So we'd like to screen the entire genome of these planarians, which is 20,000 genes. To do that, we needed to make some improvements. The first thing we did was move to a Gibson assembly strategy to generate these recombinant plasmids. With this strategy, there's no need for restriction digest or T4 polymerase reactions. And we found that if we did 2 rounds of PCR on the cDNA and overloaded the vector with the insert, that our transformation efficiency just went through the roof. We also moved to a magnetic bead PCR cleanup after the second round of PCR that was very robot friendly. Gibson assembly was then performed with clean PCR inserts and a linearized vector produced by PCR. All of this was done in a single tube in a single reaction. The Gibson assembly reaction was then added to competent cells in a 96-well plate and transformed before plating onto these 48-well segmented bioassay trays. And as you can see here, our efficiency is pretty good. We have quite a few colonies in each one of these sections to pick from. And now our colony picking robot would go into action and pick colonies, which are then screened by Sanger sequencing. Once we had this high throughput pipeline in place, we realized that we had a need for a low volume dispenser. The large liquid handling instruments are great at the jobs that they're designed to do, but there are things that they simply struggle with. Among these are dispensing small volumes into dry wells. And this is no trivial task, as anyone who's had experience with automated liquid handlers could tell you. Repetitive aspirate and dispense steps can also be a challenge, especially when you're talking about 96 or 384 sequential steps. And tip-based dispensers require dead volumes in reservoirs that is basically just wasted reagents. So as stated previously, we're doing 2 rounds of PCR in 96 well plates. The MANTIS can dispense from 50 mil conicals, as shown here, and that eliminates the need for tips. We're currently dispensing 23 microliters per well, but we're looking at decreasing this to a 10 microliter reaction in 384 well plates. We've been able to reduce the manufacturer's recommendation of a 20 microliter Gibson assembly reaction down to 5 microliters. The MANTIS enables us to dispense 3 microliters of Gibson assembly master mix into dry wells of a PCR plate. And tip-based dispensers would struggle at this. And I took a picture here. I hope that you can see this is actually 3 microliters that is dispensed right into these PCR plates, and they're all uniform. Costs for our 96 well tip boxes for our different liquid handlers vary from $2.50 to $5 a piece. And so the savings to this project over 20,000 genes would be substantial. And just some basic math here, if I throw it together, we're saving anywhere from $2,500 to $5,000 just on this single project. So this instrument would pay for itself pretty fast. So I'll conclude with a couple of interesting hits from this technique that we've been able to replicate in our high-throughput RNAi cloning pipeline. When beta-catenin is knocked down and the worm is dissected, it grows back 2 heads. When APC is knocked down and the worm is dissected, it grows back 2 tails. Now, the largest published screen, RNAi screen in Planaria, is around 1,000 genes. We're at 6,000 genes currently, and we have about a 3% hit rate. And I'm not going to share any novel hits that we've had until we've concluded the screen and published, but that hit rate is pretty solid. We have some pretty good leads here. And so just to wrap up, I'd like to acknowledge Alejandro Sanchez. He's the PI whose lab that we're collaborating with. Frederick Mann is his postdoc who's driving this work with us. Some past people that we've worked with, Beth Duncan, who now has her own lab at the University of Kentucky, Aaron and Stephanie, who are still at Stowers, and the person that I started on the screen with the ligation independent cloning, Carrie Adler, who's now at Cornell with her own lab. Andrew, he's the research technician in our lab who's really taken the MANTIS by the horn and is using that to drive more productivity with the screen. And Karen, my director here within the molecular biology core facility at Stowers. Thank you, and we'll take any questions.
Okay, so we have a question. It looks like for you, Dan, is the MANTIS integrated into any of your larger liquid handling platforms?
We don't have it integrated right now. We do have some plans to acquire a new liquid handling robot, possibly mid-year next year, and we are thinking about integrating it into that system.
Very interesting. And I have another question here about liquid classes. I think that could either be for you, Dan, or Paul. Do you have to program liquid classes for the MANTIS?
They have.
I can go ahead and take this. Or go ahead.
Yeah,
go ahead, Dan.
No, you're fine. You're probably more qualified to answer than me.
So with the positive displacement, we recommend, or we market that we're able to handle viscosities up to 50% glycerol with a plug-and-play manner. So there should be no need to calibrate it, whether you have a reagent up to that equivalent. But if you're above that, what we will do is we can slow down the timing of the sequences and then increase the positive and negative pressures to make sure that we're filling the diaphragms completely to make sure that when we do actually dispense the droplet, that is a full droplet. So short answer, no liquid classes, but if you do have a very high viscous solution, we can optimize it.
Great. So there's another question here about dispensing cells. Does the diaphragm technology shear cells?
Yeah, it's another great question, actually. So in my experience dispensing cells, we have not seen any shearing with our base settings. But once again, if you have a cell that may be fragile, we can change those parameters to make sure that the impact of the diaphragm on the solution, whether it's your media or water, is low. So then as the cells are moving through the diaphragm technology, it's not going to have a high impact. Or when the droplet actually is placed into the plate, it's at a lower speed as well. So, I don't want to say no, right? But I will definitely say that in my experience, when working with customers, it does not cause shearing.
Okay. So I have a logistical question here about the presentation slides and whether I can send them out. So this webinar has been recorded, and we will send out the recording following the event for everyone. So So one question here, how long does it take for the MANTIS to dispense into a 96 well plate?
So is that for me?
Yeah, go ahead if you know
Sure.
the answer.
So for our PCR reactions where we're dispensing 23 microliters, it takes about 40 to 60 seconds to dispense across the 96 well plate. But for the smaller dispenses, our 3 microliter reactions, it takes probably 10 to 20 seconds.
Okay, so that's great. Thanks, Dan. I have another question for you here. Have you lost any sensitivity when you reduce the volume of your PCR reactions?
No, we haven't. But then we're using we're not just loading these on a gel, so it might be more difficult to detect on gel type technologies. We're using LabChip GX and the Advanced Analytical Fragment Analyzer, so they're pretty sensitive.
Great. Okay. Well, that's all of the questions that we have time for today. So I'd like to thank everyone again so much for taking the time out of your busy schedules. I hope this was an educational event and that everyone takes something away from this. And I'll be sure to send out the recording soon. Thanks again, Dan, and thanks, Paul, for your time and for putting together a couple of really nice educational presentations.
All right. Thanks, Tom. Thanks, Dan.
Thank you.